questions about the class? Tissue sensitivity. No? All right, let's moving on. So here's where we were last time. We're discussing germ cells. We were looking at, let me go back one slide first. You can take a look and see what we were talking about. I was talking about exposing or exposure to the donuts and what happens when the donuts get exposed. And so we're saying the germ cells production in males, spermatozoa, arise from the germinal cells. And their production continues through life. One thing that you observe with male donuts that get exposed is that you don't see really a reduction of sperm until weeks after, 4, 5, 6 weeks after. Before that, you don't, because the spermatozoa are somewhat radio-resistant in comparison to the spermatogonia. Okay? But once the spermatogonia gets affected, then they are not making new sperm. So you see a big problem. All right. The stem cells go through several divisions to progress to the spermatozoa. That whole process will take approximately 10 weeks, 9 weeks. The effect of radiation on fertility in men takes several weeks, 3 to 4 weeks after the exposure. Okay? And it's relative to the stem cell. Spermatozoa are fairly radio-resistant. I mentioned that a few times. A few more things. Radiation doses as low as 10 rads, okay, can reduce the number of sperm. So we're not seeing a big dose. Let me ask you this. Is it possible, with what you know now, is it possible to deliver 10 rads in diagnostic radiography?
Yeah.
What areas?
Nuclear medicine?
CT.
CT, yes. Fluoro, not per se upper GI, very minimus. But if you're doing a cardiac cath, if you're doing special procedures, where you're doing angiograms, yeah, in that case, yes. But otherwise, very unlikely. Possible, yeah, but unlikely. We need to have several X-rays to do that. I mean, is it possible 10 rads? Wow. Is it possible to have it on a very minimal? It's possible. Yeah, it's possible. If a long case. A long case. Okay. Unlikely. Possible. But special procedures, cardiac cath, it is possible. Now, in a cardiac cath, you're not going to be over the gonads, you're over the chest. So, that's one thing. Radiation therapy, possible. But in the diagnostic range, very unlikely. But my point here is to show you that you will see this spermatogonia, it is sensitive. I mean, see, 10 rads is that sensitivity is there. Okay? I don't think we haven't seen, up to this point, I have not shown you anything as low as this, where you've seen an effect. See my point? Okay. So the threshold for temporary sterility is about 15 rads. Okay? To the testes. And so it is, those are not high doses. Okay. 200 rads produces a temporary infertility. Okay? And 500 rads will produce permanent sterility. Okay, what's the difference between temporary sterility and temporary infertility? I'm not sure. I was looking at these terms the other day and I'm going, Probably means the same. And so here, temporary means that it's probably going to come back soon after. Okay, the damage is not so great that it's going to come back soon after. With 200 rads, this is probably going to take a while for fertility to come down. Okay? And with 500 rats, that will be permanent sterility. Okay. The induction of sterility has no real physical impact. We talked about this slightly. So, anyway. All right, now let's switch to hematologic effects. This is not the hematopoietic syndrome. This is not the hematologic syndrome. This is just effects that you see from radiation, specifically on blood, okay, or blood tissue. Are you following me? Mm-hmm. Okay. Just like we look at the skin, we have looked at the testis, or we look at the gonads. Now we're looking at blood and what happens with blood. The hematopoietic or hemopoietic system includes bone marrow, circulating blood, lymph nodes, spleen, and thymus. All of them are involved in production of blood. Okay. The principal radiation effect to the hemopoietic system is a reduction in the circulating blood cells. Okay. Just like what we observe with the hemopoietic syndrome. Okay. The same thing is we're gonna observe here. Okay. Pluripotential cells, we discussed this before, gives rise to other cells of the hemopoietic system. Okay. Bone marrow. Bone marrow is responsible for the production of circulating blood cells. Okay. Especially in children, but then also as adults. Okay. You have long bones, flat bones, they're always producing blood. Okay. So in children, they are distributed everywhere, and so it is they need to be protected more than adults. Okay. As I said before, in adults, flat bones and long bones. Okay. Ribs, skull, hips, sternums, bones of the pelvis, etc. All right, we discussed this slide before when we were looking at the hemopoietic syndrome. Okay, just very quickly. So you have this stem cell, and from these cells, all of the blood cells are formed. Okay? All of them. Red blood cells, white blood cells, and platelets. They're all going to be formed. Okay. Remember, platelets are not really cells. It's just a cell, a big cell that explodes, and so these are fragments of cells. This is what you have here, the chromocytes. All right. So cell survival. The principal radiation effect of the hemopoietic system is a reduction in circulating blood cells. And so there's where you have to think about what happens. Well, if you're reducing white blood cells, well, you're prone to infections, right? If your red blood cells are reduced, then you're going to have anemia. Okay? You're not going to have enough blood oxygen being transported to your tissues. Okay, platelets, well, you're going to have problems with blood clotting. And so all of them will have an effect. Lethal injury to the precursor cells causes depletion of these mature circulating cells. Okay, and so we want to protect. What we want to do, is protect these cells, protect the stem cells. Okay, because if they get affected, then everything else gets affected. So that's the key there. Okay, ready? Here we go. Ding. Done. All right, so what we observe here, as we have seen before, right, with doses as low as 25 rads, we're going to see small changes, okay, especially in the lymphocytes, okay. So small reduction here with 25 rads, but as the radiation dose begins to increase, then their numbers begin to go lower and lower. remember, lymphocytes are very sensitive to radiation. And so not only we're going to see an effect on the stem cells, but also lymphocytes are very sensitive, and so their number is going to go down. Take a look. It doesn't look like it's much to you, but this is the number of cells per cubic centimeter. That's 1,000. So 4,000. Take a look. You see a reduction here. It might not appear like it's a lot to us right now, but it's when you see it in centimeter per 1,000. That's a big chunk. Okay, and it's going to take you what, another 2 months or so to get back to normal. So it is an effect that we have to observe. Not so much with the other ones. With granulocytes, you see this big spike, and then it goes back to normal right away. Platelets, not much of an effect. Red blood cells aren't even counted here because you don't see much of an effect. All right, any questions about this graph? No. Okay, good. All right. Cell survival, except for lymphocytes, cells in the circulating blood are considered radioresistant. And I will add here to your notes, add granulocytes. They are somewhat radiosensitive as well. You don't see the drastic change, so add yourself a note there. You don't see a drastic change that you see with lymphocytes, okay? But you do see changes, especially when you go back here, especially when you're getting into the hundreds. Then you begin to see these drastic changes, okay? And even when you get to, say, about 75 rads, you begin to see these spikes, and now, right after, you can see it deeping down. Okay, so granulocytes are somewhat radiosensitive as well, but not nearly as lymphocytes. So if on a test, on your national boards, you're asked, out of the list of cells that we have here, which one is the most radiosensitive, and you see granulocytes, lymphocytes, red blood cells, platelets, whatever. Well, you want to go with, always pick lymphocytes. That's your choice. It's always going to be your choice. If they don't give you lymphocytes, then you have to really look and study the list. Okay. You want to see that. You want to always see lymphocytes. All right, so let's go back here. So except for lymphocytes, granulocytes, cells in the circulating blood are considered radioresistant. Okay. The coincidence of damage to the bone marrow and the circulating blood cells depend on the following: the sensitivity of the various stem cells that you have in the lifespan of each cell line. Okay. In this case, it's just one stem cell. Okay, and so what we're going to look at is the different cell lines. Okay, and we observe by the cell line, it is the lymphocytes that have the most radiosensitivity. Okay, all right. Following radiation exposure, what are we expecting to see? Okay, lymphocytes are the first cells to be reduced. That's what you're going to see first. Which I talked about before. Doses as low as 10 rads will decrease the count. Remember that I had mentioned to you before that for early radiologists and early X-ray technologists, they used to do blood tests. Like every month, they used to have the blood drawn to see where was their lymphocyte count. And if the lymphocyte count increased too much, they will pull them out and keep them out of work for a little bit, or at least out of maybe not out of work, but out of being exposed to radiation. Maybe you can do paperwork or whatever. Maybe you can answer the phone. I don't. But no radiation exposure. Okay. Neutrophils are second. Okay, and remember, in the neutrophils, here's where you find the granulocytes, right? This is part of them. Okay. Doses as low as 50 rads will decrease their count. Platelets and red blood cells are the last to respond. They do respond. Remember, any tissue, when exposed to radiation, is sensitive. It's just that they have different levels of sensitivity. If the radiation dose is high enough, all tissues can be affected. There's no no exception. Okay? Every tissue will be affected, but in this case, the last to respond are going to be red blood cells and platelets. Okay, and here, for you to begin to see changes, you need doses higher than 50 rads. Typically, you won't see any drastic changes with doses lower than 50. All right. Cytogenic effects. All right, the study of the genetics of cells, in particular cell chromosomes. So now we're going to look at what happens to chromosomes, what happens to genes, etc. Let's take a look. So radiation aberrations follows a non-threshold dose response, and it is related to dose received. So what is that telling you? Aberrations follows a non-threshold. What is that saying?
That means that any level of radiation.
Any level of radiation, okay, as low as it can be, it is suspected to have some kind of effect on the cytogenic system or the cytogenic tissue. Okay, so radiation can replicate any type of aberration known in nature. So anything, any changes that we see with genes, any defects that you might see with genes, the radiation can replicate it. Okay, and I mentioned to you this before as well, that radiation will not make anything new. It will not make any new aberrations that we don't see in nature. It just doesn't happen. But it can replicate any of them. There are some that are more common than others. But for the most part, you don't see it. Okay? You don't see nothing new. All right, so when we look at genes and genetic material, how is it organized? First you start with something very small molecule of DNA. Okay, it's a tiny molecule. And so if we go up, when you expand it and you combine DNA, you have groups of DNA that is called chromatin. Okay, chromatin, if you group them, okay, then you have genes. Okay, and if you have a bunch of genes all combined, now you have chromosomes. Okay, so this is pretty much the order in size, how they are organized. So you start with, so DNA is you can say is the basic unit, and then chromatin, genes, chromosomes. Okay. Yeah.
Chromatin.
I'll explain each one of them in just a second. Okay. You got this picture? Okay, good. All right, so DNA and chromatin. All right. Chromatin is composed of nearly equal amounts of DNA in globular histone proteins. Histones provide a physical means of packaging the DNA molecule in very compact and orderly way. Plays an important role in gene regulation. Okay, so this, when you see this compartmentalization of DNA, that's chromatin. Okay, it's a group of chromatin. A solenoid is a chunk of them, but don't get too involved in that. I just want you to know or remember, DNA is set into these compacted structures called chromatin. All right, so levels of DNA condensation. So the first one is the DNA strand of helix. Okay, now you have a chromatin strand. See these little groups here? Those are chromatins, just like I was showing you before. Okay, condensed chromatin during the interface of the centromere. That's all of these. Okay, number 4 is going to be condensed chromatin during prophase, and then last you're going to see chromosomes during metaphase. Okay. All right. Microscopic pictures. It is difficult to appreciate details of chromosome structure even with an electron microscope. So here what you have is just a whole set of condensations of, you know, start with DNA and then genes and then chromatin, and it's all coiled up, all right? And so, and that's what I had referred to chromosomes as condensed miles and miles of DNA, okay? Because it's just a coil that has been condensed into this small area, okay? But even with, I mean, just to, why I might have this note here is just to show you that even with a with an electronic microscope, it is difficult to, or electron microscope, it's difficult to appreciate. It's not as easy as people might think that, oh, I want to look at my genes. It doesn't work that way, okay? It's difficult to see it, okay? All right, however, one label them with dyes that are preferably taken out by certain regions. And that's, you do these dyes so you can, so the chromosomes can take them, and then you can see bands of different colors, because different areas of the chromosome will absorb more or less of the dye, okay? So if you have seen pictures, or if you, maybe a biology class that you have taken and you saw chromosomes and they are stained, that's what they were showing you, okay? It's just a dye chromosome. They added the dye. Different areas of the chromosome absorbs the dye in different amounts, okay? So, and it's just so you can create this banding pattern that can cause, that can help you identify individual chromosomes. All right, going back to the way it is organized, okay? So if you start here, okay, with a DNA helix all the way down here, and the histones here, okay, is just a little packaging of the, where you have the DNA and the chromatin there. And these histones keep on packing, and when you have many of this coil thing, then you end up with a chromosome, which is what you have through a metaphase. So take a look at that. This is just different levels of uncoiling. Just to give you a sense, once again, of how small they are, 4 nanometers DNA filaments, okay, the DNA helix. I mean, that's nothing. I mean, not physically nothing. I'm just saying how small they are, okay. Double-stranded DNA wrapped around sets of 8 histones to form 10 nanometer filaments. Okay, so very small structure. Sets of 8 histones wrapped by DNA are separated by space regions of 4 nanometers. This is spacing that you see here. All right, you can take a look at that. Okay, this is just to kind of help you remember a little bit about chromosomes. Okay, each chromosome contains many genes arranged in specific linear sequences, constricted at certain points by a centromere. Remember the centromere right there, okay? A clear region necessary for movement of the chromosome during cell division, because the chromosome is going to be moved through because of the centromere, right? Remember, you have the spindles. Remember that? The spindles come through the centromere. So this is how the centromeres, this is how the chromosomes move, and so they need that centromere. Chromosomes are constant in number for each species. How many for humans?
46.
46.
Mm-hmm.
All for
23
46,
pairs.
right? Huh?
23 pairs.
Yeah, 46, right? Okay, good. Okay, here again is just to give you a sense of how small these things are. So, okay, DNA, the double helix, 2 to 4 nanometers, beds of the strings to form a chromatin, about 10 to 11 nanometers. I mean, what's the difference between 10 and 11 nanometers? Don't. Okay. This is a section of the extended chromosome. It has been stretched. It's about 30 nanometers. And when you get to the chromosome, this will be about 1400 nanometers. Okay, the entire thickness of the chromosome. All right. Okay, good. Need it more time? Huh? Yes?
You did say 300 nanometers, right?
Yes.
Okay.
Right there.
I thought you said 30.
No, they're 300. Yep. That will be a big difference.
Oh, yeah.
1 nanometer is not big. At least from my point. All right, chromosomal aberrations, often caused by breakage of incorrect rejoining. So why am I telling you all of this about chromosomes? So you can get a sense of where and how radiation will affect it, okay? And how small. Remember when we were looking at point mutation or point lesions, and we were looking at ring breakage or ring formation. Now think about it, how small it is and what areas of the chromosome is really being affected. So when you talk about point lesions, you're talking moving individual bases. So now it's something really tiny. You're talking about 2 nanometers, 4 nanometers. So it's something very specific that happened at the molecular and atomic level. Okay, it's not something that happens at 1,000 nanometers. It happens at very specific locations, okay, especially with gamma and X-ray radiation. Okay, so keep that in mind. Get a sense of the size and where the damage will happen. So broken segments may remain separated from minutes to hours. Okay, and at that point, when pieces of the chromosome are broken, the end pieces are said to be sticky. Okay, and here's where you can have ring formations. A lot of times because they are sticky and they want to combine something else. That's not the way they were supposed to be. And so they are going to find a way. They stick to the chromosomes. They can stick to other areas of the chromosome itself. Make sense? Okay. Right. They are capable of reattaching to any other broken segment, most often rejoining in the original configuration. Hopefully, that's what happens. Hopefully, that's what you want, but why not? So dose rate effect for single strand breaks. Okay, lower dose rates allow for greater probability of repair. That makes sense, right? The lower the dose, yeah, you have a greater chance of repair. The greater the dose, it is very difficult because it might not only be one strand that is broken. You have multiple areas broken. And the same thing can be said if we talk about different types of LET radiation. You're going to the high LET radiation. Now you're not removing one small piece, one small portion, and now you're removing chunks, areas, whole large areas of the chromosome. And remember, when you say, well, that's really not much what you're removing of the chromosome, but now those are miles and miles of DNA, isn't it? Okay? All right. So, provided there is sufficient time for the single strand break, underlying single strand to be repaired prior to the formation of a double strand break in the vicinity. All right, thus, 2 neighboring single strand breaks identical to a double strand break. Less likely to happen, but possible. Okay, possibility of a threshold dose rate. Well, maybe, maybe not. I think it has to do, obviously it has to do with the dose. And I'm talking with the energy of the beam, the dose that has been delivered, and at the state in which DNA is at the time of the exposure. If it is during metaphase, yeah, okay, maybe there is a threshold, but not enough information to say that there is a threshold. So for now, we're going to think of effects of radiation on DNA or effects on genes to be non-threshold. So put yourself a little note here, what I just said, possibility of threshold, and then what I just told you.
No,
And I'm not going to repeat it. Pay attention. Think about it. Okay. Radiation-induced induce aberrations. So 3 major lethal aberrations that we observe. Okay, one is dicentric, ring formation, and anaphase bridge. So and 2 major non-lethal aberrations. Okay. What is lethal, by the way?
Deadly.
Okay, deadly. There you go. Okay, good. Just to make sure. All right, 2 major non-lethal aberrations that we have: translocations and deletion. Now, when I said that is non-lethal, I'm not saying that there is no damage. Okay, and I'm not saying that there will not be any consequences of the aberration. Okay, there could be consequences, especially with translocations, and with both, with deletions. If you remove something that is important, yeah, you're going to see effects, right? It might not be deleterious, it might not lead to death, but it might be something serious. So just because I'm not saying that it's non-lethal, it doesn't mean that it's not damaging. Two very different terms. Okay? All right, is that clear? Okay. So let's take a look and see what they are. Example of dicentric, and dicentric will be lethal. Okay, please lethal. The steps in the formation of dicentric and an acentric fragment. Okay, so what you have, what you have here, so you have 2 different pre-replication chromosomes. So no replication yet. So you have these 2 chromosomes. Let's call them chromosome A and chromosome B. So what you have, take a look. Here is the centromere, here's the centromere. What we observe is that radiation comes in and boom, breakage here and breakage here. Okay? So now, remember, the 2 ends that got broken, they become sticky. All right? And so what happens is you will end up with this type of formation. With this piece of this chromosome will attach to this long piece. So the 2 long pieces will attach, and these 2 short pieces will attach. No centromere there. And so you have, what you have here is this dicentric chromosome. Okay, 2 centromeres, 2 pieces there of the chromosome. And then you have these 2 areas here, this is after replication, right? Then you have dicentric chromosome plus the acentric fragments. There's no centromere in these 2 fragments. Okay, so that's lethal. Right? We're done? Okay. Now let's take a look at ring formation. Okay, so take a look. So this is pre-replication chromosome. Okay, centromere. Now you have a radiation exposure. Okay. Or, by the way, not only radiation exposure, chemicals can do this too. There are many chemicals can do it. Free radicals will do this too. Okay? So it's not only radiation. I mean, radiation is what we're studying here, but many other agents can also do this. Okay? All right, so you have 2, 1 breakage here, 1 breakage here. Okay? And what you have is you have these 2 sticky parts. Okay, these 4 sticky parts actually, and so these 2, they combine, and this is an incorrect union. You don't want that. And then you have this acentric, once again, this eccentric fragment. And then after replication, what happens? You have 2 rings. Okay, and that's not the way it was supposed to be formed. And then you have these 2 separate fragments. Okay. All right. Now, bridge, okay? Right, once again, this can be lethal. So what we have is here. So we have this chromosome here. And take a look. Everything that I had shown you before was pre-replication. Now take a look here. This is post-replication chromosome. All right, so what we have here. So we have this chromosome nicely formed, well done. Now radiation comes in, boom, breaks it. And now what you have, these 2 pieces just boom, combine, okay, and these 2 pieces combine. This fragment is acentric, okay, and what you have, okay, you will end up with, this is a big problem, and then you can end up with 2 dicentric chromatids after you have done more replication, an acentric chromatic fragment. So, big problem there. Rearrangements, okay, rearrangement can be non-lethal, and however, just because it is non-lethal, it doesn't mean they cannot lead to bad things. And so it is involved, and this special rearrangement is involved, or this special effect is involved in carcinogenesis. Okay. Translocations. Here's where you have breaks in two chromosomes, and they translocate. They send one piece of one side of the chromosome goes to one side, and the other piece goes to the other side. Okay. The sticky ends are exchange, deletion. Okay. This is when you're removing a portion of it. Did everyone okay with translocation? What I said? So you have a piece of chromosome. Centromeres. So you have a piece of the chromosome, say this area here and this area here. They are broken and they come apart. And then this piece and that here, and this piece and that here. Okay, that's translocation. Which, by the way, happens. It happens when during fertilization of the egg, there is an exchange of genetic material. But that's let's say prearranged. That's predetermined that it's going to happen. In this case, it's not. It happens because of radiation. Deletion. Here's where you have one break or multiple breaks sometimes of a chromosome. Okay. Information between these two breaks is lost. It's gone. This is when radiation will come in, and boom, you lose the whole portion of the chromosome. Okay. So that information is gone. Might be good, might not be good. You know, typically it's bad because here you have miles and miles of DNA in this arrangement here. And so all of that is gone. It's lost. Okay, so symmetric translocation, non-lethal again. You can see him a little bit better. All right, Dennis, you had a.
I'm sorry, I was hoping if you can go over the dicentric once more in the ring lethal example and the bridge.
All of them?
Just, or
right.
maybe I can
let me
just
go back.
verify.
All right, let me go back. I'm sure it's not the only one. All right, so let's take a look. So let's start with dicentric. Dicentric means what? Di's two, right? Two centers. So that's what you have. So you're going to have two centers. Why is that? Because what is important here is this happens before replication. That's why it's pre-replication chromosomes. All right. They're normal. They're fine. Radiation comes in. Bing, bing. It's both of them. Okay. What you're going to have, this is sticky end. Remember what I talked about, sticky ends? Okay, this is sticky end. This is sticky end. They just combine.
Is that translocation?
No. This is sticky ends, they end here, okay? And this is sticky ends, they join. And so now what you have is this chromosome with two centromeres. So that's why it's called dicentric, because that's what you end up forming. And then the problem is, it doesn't stop there. The problem is now you're going through replication. And now they are going to replicate the mistake. That mistake is not fixed. All right? So now you have this chromosome, this malformed chromosome. Okay? Two centers, dicentric. Okay? And then in addition to it, you end up with two fragments that don't have a centromere. If they don't have a centromere, what's going to happen? They just don't move. They don't go anywhere. Okay?
They won't reproduce, so they won't become cancerous.
They cool. Yeah. That cell that keeps that will lead to. But well, remember, it's lethal. So will it lead to cancer? It might, but the tissue might be already be dead. It might already be gone. Okay, whatever it was leading to. Make sense? All right, another one. Ring formation. So here you have, once again, it's a pre-replication chromosome. Okay, it's a full chromosome. You have a centromere there, centromere here. Radiation, it breaks that chromosome. And what happens is you have, once again, the sticky ends. they want to combine with something. Okay, a lot of times they go back and combine with the broken fragment where they came originally from, and that's good. A lot of times they don't. They just combine. This one will combine with this one and forms a ring. Okay, and that doesn't happen in nature. You don't have chromosome rings. Okay, it just doesn't happen. In addition to it, you have a fragment with no centromere. Now you go through replication, and what you have is now you end up with two rings. Okay. All right. So that's a problem. And then also this is replicated. Okay. All right. Now bridge. What happens with bridge? So you have now here's key with bridge. Here this is post replication. The other ones that I talked about was pre-replication. When does replication happen?
In the synthesis stage of interphase.
Okay. And where is that in relation to mitosis and meiosis?
After growth 1.
Huh?
After growth 1.
Or before growth 2.
Right.
Oh, okay. Just checking. Okay. I want to be talking to people who don't understand that. All right. So, chromosome here is fully formed. Okay. Okay. After replication, okay, radiation comes in, beam, it breaks that chromosome. Okay. Now, what do we find? Is that these two pieces, they combine. Okay, and okay, this is not something you see in nature either. Okay? This is abnormal, badly abnormal. Okay? And it will lead to death. Death of what? The person might die? Well, maybe not the person. Okay? But the tissue, what it was leading to. Remember, all of this is DNA. And what is the purpose of DNA? Everybody?
Instructions for the cell to reproduce and to create its function.
And all the functions of cells are directed by DNA. And if you don't have that DNA, or if the DNA code was messed up, what's going to happen? The functions that the cell is normally doing is not going to do it. So that cell will die. Okay? Remember, always think, because a lot of times we look at a very abstract fact, but it's not. If DNA is not there, and that DNA, that code was important for something else to create a protein or reproduction or whatever, and it's not there, then the cell will die, okay, if it is something important, okay? Imagine if it is a code that makes, I don't know, ribosomes. Now the cell doesn't have ribosomes, okay? Or maybe it is a code to make cell membranes. Now you don't have cell membranes. See all the problems that it brings? Okay, so this is when you're talking about, especially when you're talking about removal of chromosomes or pieces of chromosomes are being deleted, that's serious because you have multiple sets of DNA. Make sense? All right, okay, good. So this is what you have. So this is not well done. All right, okay, let's move on. Rearrangement, not lethal. We're talking about that. Where are we? Here. All right. We okay? Dennis?
Yes.
Everybody else?
Thank you.
Yes. Everybody else? Because I know it was not just Dennis. Don't give me that. Yes.
Yeah, but I just summarize. It means Dicentric and the
You
ring.
heard it from
No,
just to summarize.
Yeah.
Tell me.
Yeah, for me, I'm not sure I correct. Dicentric and ring is happen at the Z1.
What was it again? Say that again.
It's a dicentric and ring is happen at the G1.
Yes.
Yeah, and the break it happen at the anaphase.
Yeah. After no. After duplication.
After is the G2
After
or
synthesis. Fully happening. That could be in G2.
G2.
Could be. Yeah, because it's after duplication. Okay.
Yeah, thank you.
After S1.
All right.
After.
Okay. So what we have here is pre-replication chromosomes, okay? So nothing has happened, so they haven't replicated yet. So we have here radiation-induced breaks in the adjacent chromosomes. So you have radiation comes in, radiation comes in. So you have problems in adjacent chromosomes here. So what happens after that? What we observe, okay, is that a portion, and I don't know why this didn't come out green. Imagine this is green, okay? I don't know why it came out this way. Or this is blue and this is green. So a portion of this chromosome, but it now is not only a small piece. These are chunks. These are whole legs of the chromosomes. This chromosome here, look at this one here. This one here? Okay, now it's here. So paint it in your thing, paint it green. Okay, and this blue, okay, this portion of this chromosome is here. And remember, DNA in chromosomes, DNA is not randomly arranged. This is not like, don't think of, a student a couple of years ago said, well, are chromosomes like when you're packing DNAs, like you're going on the train and just people going? No, it's not like that. They're nicely organized nicely organized in a way that the function that they are going to direct are related. Okay, so it's not a disorganized arrangement of DNA histones. Okay. That's why you have sex chromosomes. Remember those? Mm-hmm. Yeah. And you have different chromosomes that do different things. And so DNA is organized depending on the function they are going to do. Okay, Dennis?
So would that cause mutations and deformities?
This will cause a lot of problems.
Or...
This will cause a lot of problems.
It's not death, but it's still
It is a lot of problems. One of them is cancer. Okay, depending on what chromosome is hit. Okay? Make sense? Okay. Yes?
What makes it symmetric, just they're both from the
Oh,
plot?
no, it doesn't have to be symmetric. This is just it's just a name. Don't think that it's going to be perfectly aligned. No.
But this is a type of translocation?
Yes, it is a translocation. Yeah, it is a translocation. Okay. All right, broken pieces are exchanged. So these two pieces they're going to be exchanged. And by the way, in addition to this exchange that happened, there are pieces missing too. There are going to be pieces of DNA that are going to be gone forever. Okay, bases that are going to be gone. They will pulverize by radiation. Gone. But and the pieces that remain, they are going to join, but they are going to join where they belong to. Okay, all right. Not necessarily lethal to itself. Okay, may lead to cancer because of loss of suppressive genes. Remember we talked about suppressive genes before? Yeah, I did. You don't remember that? Suppressor genes? Suppressor genes, that's the name is saying. They suppress the induction of cancer. Okay, that's their function. They stop the development of cancer. Right? That's suppressor genes. Maybe I didn't talk to you this. Okay. All right, small interstitial deletions. These are going to be non-lethal. Okay, pre-replication chromosome here. Now you have radiation. Radiation-induced addition breaks in chromosome. Ping, ping. Okay. And what you have, fragment loss at mitosis. This piece here is gone. Disappears. Okay? Not necessarily lethal to the cell, but it can cause a lot of problems. May lead to cancer because of loss of suppressive gene. Okay? See this piece here? Okay. And instead of being long like it was here, now you have the short little tail here, and this piece is just floating there. Okay? All right. All right, let's stop here. That's it. By the way, we're almost done with this section, just about done. So what we'll do is when you come back, the week you come back, you're gonna have Tech 430 is on a Monday, okay? And Tech 415 will be on that Friday when you come back. Right, come back.
Wait, test or quiz?
Exam. Exam. We're done with this section.
3 exams and
Yeah, but we're gonna be done, that's what I'm saying, guys, we are going to be done.
It doesn't include the chapter 34 that we have,
You will include this whole section,
and that's it, okay.
that's it.
So this is the last
So no
slide.
vacations.
Well, no, when we
Okay, I
you're
agree.
right, I've just I'll put it, just look at your web access, just look at the dates.
Okay.
Because we're gonna be done Monday and Wednesday, we're gonna finish, and what I'm saying is get ready for Friday.
Okay.
So, that's what I'm saying. Make sense?
Yeah.
Okay.
Let's get it.
And if we're not done by Friday, by Wednesday, well, we'll move it, okay? But what I'm trying to say, study.